talk about this a little bit. So this basically is this is showing you the spectra of white light. And so if we break up white light, we can sit there and get, you guys learned all this in chemistry. You should remember this since you both are here. But if we break up the spectrum, we get all of the different colors, right, of white light. So we get the violet, the blue, the cyan, the green, the yellow, and the red, okay? So, so there are basically, our eyes are limited. And so initially, when we think about the rods in our eye, there are three types of rods that exist. They're able to recognize certain pigments. They're going to recognize, we have blue cones, we have red cones, and we have green cones. Okay. And then let me elaborate. There are three types of cone opsins. Okay, and so we're going to recognize red, blue, and green. Okay, and so we're going to use the percentages to kind of sit there and get those distinctions of all of the different colors. Okay, and so this here just basically just shows you the spectrum where those cones are being recognized. Like I said, we have the red, the green, the blue, the red, and the green. And then our rods is going to be the combination, getting pretty much that full range. Okay. All right, you with me so far?
Yeah.
Yes.
Okay, so okay, give me 1 second. Okay. Okay, this is going to be the ascending pathway of the spinal column. I'm going to try to put some of all the features. So this guy is going to be a third order neuron. This is the post-central gyrus. Okay. This is going to be your second order neuron. There. Down here, you're going to have your first order neuron. Okay. And you can see the image where it actually says spinal cord. I know the images are really hard to see and kind of make out, so trying to put them up there so you kind of get the idea of where they're at. They're actually in the book, and they show it a little bit better, but they're still kind of tiny in the book. As I get older, I realize that my eyes get worse. Okay.
Yes.
So basically, we're thinking about the sensory, the peripheral sensory are going to be going from our spinal column up to our brain. Okay, and so we're going to actually take the route through the spinal column. So the major path that is going to be ascending from the periphery to the brain, you're basically going to go to your spinal thalamic tract, and basically we see the thalamus here. So we're going to go along that spinal thalamic tract, and then we're going to be coming up from, let's say we get some type of touch or something along those lines. So we're going to go from that spinal cord or to the dorsal root ganglion up to the up through the follicus contius and the follicus gracilis. We're going to continue up. Then we're going to go to the nucleus contilius, and then that's going to take us in this direction through the nucleus gracilis, and then that's going to go to the neo lamicus up to the thalamus. Okay, and so then the third order neuron is basically going to take us to the postcentral gracilis. So, and that's going to be like a fine touch. And then in terms of pain and temperature, so we're going to go to that first order neuron to the spinal column. Then that's going to take us to the dorsal root ganglia. It's going to go through the lateral spinal tract. And then that's going to hit us to the second neuron. Then we're going to go to the third order neuron, and then that's going to take us to that post-central ganglius. So depending on the path or the sensory that we're taking a look at, that's going to sit there and send that signal through those distinct paths. So in one case, it's going to be that dorsal column system, and then in the other case, it's going to be the spinal thalamic tract. Come on. Okay. So this basically is this is going to be the auditory brain system mechanism of sound. It's where it's located at, and so if the source of sound is to our left, so the ear on the left is going to be the one that hears it first, right? And it's going to be louder. The sound on on our right, I mean the ear on our right-hand side is going to arrive a little bit later, and it's going to be softer. Okay, and so it's the neurons on both sides are gonna compare. You know, they're gonna be able to compare those slight differences between stimuli based upon where they arrived at and when they arrived. Okay, and so they're going to recognize that difference between intensity. So then naturally, so we hear from here, our response is going to be to look in that direction because that was the one that we heard most from, and we know that it was a little less on this side. Okay. So if the sound waves were coming from straight forward, then they're going to have the same intensity. And so then in that context, we're going to be looking straight forward because we recognize that the sound is coming straight ahead from us. Okay. And so it's going to do that comparison and recognize that they're at the same level. Okay. Questions, concerns, Cash?
So it just depends what angle you're I mean it just depends what angle the hearing or comes from.
Yes. So and again, and that's it kind of makes sense because the source, if the source is towards one side or the other, right, you're going to have a lot more waves that are impacted, so a lot more pressure on one side compared to on the other side, right? Because it's coming from this direction.
Yes.
Okay, so when we talk about this, so your brain is actually looking at, it's called the inner auditory time difference. So when those signal arrives, so your brain is calculating that. It says, okay, and then it's also calculating the inner auditory intensity. So the how intense it is too. So it's looking at the differences between them. So we're looking at the time and the intensity, the inner time and intensity differences between the two. So we're able to sit there and compare those. Okay, so in terms of our vision, so our eyes are going to be moving, you know, so in the context of when we're looking around, so our eyes are going to be constantly moving. So if our head is moving, we're going to kind of compensate for with eye movement as well, right? To help with our visual. So we can sit there and see things. We can see that visual stimuli, even though our head is moving in a particular direction, right? And so there's going to be a compensation as a result of eye movement because our head is actually moving. Does that make sense?
Yeah.
Yeah.
Okay. And so, you know, that kind of helps us, you know, dictate, you know, it's like, okay, so even though we're focused, you know, our head is moving back and forth, I can still see those visuals. And so our visuals, our eyes are compensated by them moving independent of our head moving to do that rapid connection. And so those compensations are still sending those signals down down through the neurons to our central nervous system or our brain. Okay, and that, let me go back. So, and this pathway is called that it's actually sending these signals through. It's called the vesicular ocular. It's called the vesicular ocular reflex. But it's that pathway, the vesicular ocular pathway, that the that is being used to sit there and send that signal or that reflex down. And so this basically is just showing you how things are processing. And so we have the signals that are being sent to the right side of our brain, and the other part that's being sent to our left side of the brain. Okay, and okay. Okay, and then if we take a look, one side is representing the excitation, which is going to be in the red, and then the other side is representing the inhibition. And so one is recognizing that excitation, and the other side recognizing the inhibition. So anybody who's had like a traumatic brain injury, and let's say part of their cranial structure has been damaged. So if one side is damaged, we could sit there and not receive certain stimuli. So things that may excite us may not excite them, like visually, as a direct result of it. Where they may get a response to some type of inhibition, but nothing in terms of the excitation. Does that make sense? So they may be able to respond to being stimuli that exciting the eye movement, like a flashing light. So they may not be able to respond to that, where they're still able to sit there and close their eyes, or they're still able to sit there and I'm trying to think. They still may be able to respond to, let's say, the sun going down, right? They recognize that, where they're not getting any excitation, where the light is shining. As soon as it shines in their eye, they're not being excited in that sense. And so there's no response. So their eyes is not dilating as a result of it, because there's no response in that regard. Does that make sense or not?
Yes.
Okay, so like one of the ways that people determine whether you're high or not is by your pupil dilation. Okay, so and that's a response to your pupils start to dilate as a response to, let's say, marijuana intake or a certain type of drug intake. So then you get a result of that. Right? So in this case, you could be smoking marijuana and your eyes won't dilate. Okay. So if you had damage to a portion of your brain. Okay. Okay. So in the case of your visual field, so in this figure, basically just showing us that, so we have the right side and the left side again, and in these fields, right, so this here is our binocular fields, and so we're going to have overlap between the two sides of our brain. So this guy is going to be for the right eye. This guy is going to be for our left eye. Say. And okay, I'm just kind of getting hung up on the sides of the images. So our right hand side is really my left hand side. So this here represents the right eye. This is going to represent the left eye. And so, so images are inverted. So just a little, making sure I'm not missing something that I need to tell you. Okay. Okay. Okay, so what we're seeing on our right-hand side is going to be projected. It's going to be projected to the left-hand side of the brain. And what we're seeing on the left-hand side is going to be projected to the right-hand side of the brain. So the opposite side. And in regards to our field of vision. And so you're going to end up with that crossing of that information. So this information is going to be processed and it's going to be delivered to the opposite side than the other information. Okay, and during that process, that crosstalk where, excuse me. So we're recognizing, we take a look at our eye. Our eye is going to be able to sit there and process. Okay, right side to our left side, and it's going to take that information and process it on respective sides of the brain. And so it's going to be correlated so that all of the things that are going to be on one side is going to be sent to that. Things are going to be sent on the other side. So single eye recognizes 2 points, right side and the left side. Does that make sense? So the things that I see, let's say I close one of your eyes, right? And if you look, you can still see things on the right-hand side. And so that is being processed on the opposite side, those things that are being here. So it's going to take that divvy line. It picks up signals. Okay, so this is my divvy line. I'm going to go ahead and process this. And then I'm going to take the other side and process that. Okay? Mm-hmm. Okay. So each eye has the ability of doing dual processing. So it sends through those neurons or those nerves, those optical nerves, it's going to be sending the information and divvy up the information to where it belongs. So you'll have 2 sets of optical nerves, one on each side of the eyeball, that's going to recognize that full field of view, okay, or that full visual field. Okay. Okay, so this is so this here is just basically a portion of your brain, and it's actually recognizes the it's just showing you what aspect is controlled by which region of the homonicus, uh, the sensory harmonicus, okay, and of the cortical region of your brain. Okay, so if you gotta kind of take a look at it, you know, we have, so our toes and our genitals and stuff like that are going to be located on these leaves here, and then, you know, so basically from our feet up to our shoulders and heads, and then we can start thinking about our fingers and things like that, and then here it represents the face, the teeth, and all those things, right? So again, this cartoon just kind of gives you an idea of where these the sensory signals are going to be delivered to, and so where they're going to end up in that region. Okay, so this term here means little man. And so if we sit there and we think about that cortical processing, that whole region there is that cortical region there is responsible for receiving some of the receptors from all of the different regions that make up who you are, all of the different components. So it's receiving all those signals from there. Come on. Okay, so this here is this is more topical mapping of the retina. And so in the cerebral cortex, we can kind of see the breakdown. So the superior left visual cortex is going to be, so it takes that image, it flips that image, and then it processes that images in the cortex region so that we can actually recognize it. So if we take a look at the image, we break up the image into components. This is the upper field, this is the lower field. This is the right side, this is the left side. So it breaks up those components, and then in different parts of the cortex, we basically put those components together. Okay, so the superior left of the cortex is going to sit there and recognize that left component, right? So if we, or the opposite side of that, so it'll recognize the upper component, so that'll be the right upper field, and then the inferior left is going to recognize that lower upper field, and then the superior right is going to recognize the upper left field, and then the superior left is going to recognize the upper right, the lower left field. Okay, and so again, it puts each of those components together, and then it gives us that image. Questions, concerns, Cash? Mm-hmm. Okay. Okay, so when we talk about like depth perception, our eyes are able to sit there and extract the information about like how far things are in, right? And so different spots on your two retinas are being used to sit there and get a rough estimate of how far something is from from you, right? So for instance, if you have your finger right in front of you, right? Both your eyes, each of the retinas are extracting that information, right?
Yeah.
If you do me a favor, stick your finger in front, like simply in front of you, and then cover one eye and take a look. And then stick your other finger in front of you, cover your eye, and then take a look. Do you see a distinction between that depth perception or not?
A little bit.
Okay. So if we look at something further away, do the same thing.
When it's a much wider picture, I can see, I guess, everything that the other eye sees.
Yeah. Yeah, I mean, I can see everything, but in terms of how far things seem, like for me, my right eye, it seems closer to me with my right eye than it does seem with my left eye. My left eye makes it seems like it's further away, where my right eye seems like it's a lot closer, like, you know, to the point where I can almost touch, you know, where my left eye makes it seem so far more distant.
Okay.
Okay, but when I put them together, I get an adjustment between the two.
Mm-hmm.
So when the information is being processed from both retina, I'm getting that adjustment.
Okay.
Okay, so like, have you guys gone to a movie before and used the 3D glasses?
Oh yeah.
Yeah.
It hurt my eyes.
Yeah, yeah, a lot of people get headaches from it. Okay, so because they're playing with the fact that each of your eyes are compensating, they're compensating for that disparity. And so like one is overshooting it and one is undershooting it. It's that there, it allows you to get more, because it plays with that and it intensifies that. So if you notice, like one side will be a red and one side will be a green or something like that, you'll have two different sides. And if you just look at one side, it overcompensates on one side and undercompensates on the other. So it makes the image more 3D to us.
Okay.
Okay.
Yeah, I always wondered about that. Like, how is it that, like, you know, one side is red and one side is blue, and we still are able to see colors?
Yeah. Yeah, and that's the reason why, you know, it just, all it is it's augmenting. The filters are being used to augment that distance. That distance seems a little bit longer than on one eye than on the other, you know, and then it one is further away, one is, you know, so it kind of amplifies it, giving it a more three-dimensional sense to it. Okay, let's see. Okay. Okay, so this basically is just showing you kind of the streams. So if we break down our brain, so here's our frontal lobe, our temporal lobe, our frontal lobe, and our occipital lobe. So in the case of the stream where, so what is it, to kind of recognizing visually, what is it that I'm seeing, right? So that's going to start from our occipital lobe. Actually, they're both going to start from our occipital lobe, but it's going to go to our temporal lobe. So that's going to give you that information. Okay, so here, this is, what is it? Okay, so it's a deer. Okay, I recognize that it's a deer. Now, where is it? That's going to be recognized here. It's going to go from our occipital to our dorsal stream. So, I mean, sorry, our occipital to our frontal. Okay, so in this case, this guy is going to be our dorsal visual stream. And then from here, from our occipital to our temporal, that's going to be our ventral. And so if we're having trouble determining where it is in terms of, like, say, distance and stuff like that, we may be having some issues on the dorsal component of our brain, where dorsal, and then where if we're having trouble making out what it is, right? And so we know that that is probably dealing with the ventral part of our brain. So remember, so I talked about head damage.
Yeah.
Okay, so there's this guy that had this, and we learned about this in like 5 different bio classes for 5 different reasons. But so there's this guy who basically had this traumatic brain injury. He actually had a rod sticking through his head, as Fellinius gauge. And so it basically, it had impelled the prefrontal cortex of his frontal lobe. And so I should say, and then so after the accident, he had personality changes. And let's see, and part of that, it was, let's see. So and then he also had issues with memory as well. And so it says, let's see. So before, like, they said that he was a really hard worker and stuff like that, and he was really friendly guy. Then he went from being basically being very friendly and amicable, he became very irritating and temperamental, and he didn't want to go to work. And so they so they often would associate it with, like, oh, he must have been drinking, or maybe he's using drugs or something like that. And so they use that as means of suggesting that that effect can be kind of related to the same thing as, somebody who's abusing drugs,
Mm-hmm.
in terms of alleviating the pain. Okay, so this basically is so this is just the major descending tract. So the cortical spinal tract. So this is just basically the tract that's going to be controlling our skeletal muscle movements. And it's going to be composed of 2 types of neurons. You have the upper motor neurons and you have the lower motor neurons. And so if you take a look here, you have your lower motor neuron being shown at the very bottom here. And then this here represents the upper motor neurons, the neurons that are along this bundle. Okay. So the upper motor neurons, they have its cell body basically in the primary neural cortex. And then the lower, let's see. So it's the primary neural cortex of the frontal lobe. And then the synapses are going to be located at the where that neuron is basically going to meet in terms of the spinal cord. And so you can kind of get an idea of that. These neurons, I mean, they're pretty long in their length. And then and these guys are going to be projecting it's going to be, these guys are going to be sending those signals down to the skeletal muscle in the periphery as well. So, and I believe that is pretty much it for this chapter. I'm trying to think if I missed anything that I should tell you about. So there are going to be other connections that are going to be extending from the brain to and the spinal cord, and these guys are going to be called extrapyramidal systems. And you kind of see the pyramids here. So they're going to be basically they're going to be extending from that pyramid regions in the medulla. And then we have, let's see. Okay. Chapter 16. Give me 1 second while I pull up the next set of slides. Okay, so next we're going to be looking at is the fight or flight response, or I should say, let me take a step back. We're going to be looking at the autonomous nervous system. Okay, so here we're going to be taking a look at, kind of those kind of instinct type of things, right? So so one of the major things that we know about the autonomous nervous system is it kind of triggers that instinct. Whether when we're faced with a threat, are we going to stand and fight, or are we going to take off running and run for our lives? Okay. So, today we're not like necessarily being hunted by like lions or tigers or dinosaurs, right? So currently we still trigger these responses, but to different types of stimuli. So prior it would be based upon a predator coming after us. But with the modern day, it's not that. It's dealing with, just walking down the hallway and somebody saying, "Hey, Henry, you're supposed to get something else done." So that's going to signal some of those same responses that I had when, let's say a lion was going to be coming after me. Shit, I was supposed to get that done, and now I have this pressure. Unless I really didn't care about the project at home, right? So we're going to be talking about how the nervous system kind of deals with those type of stimulus. Okay, and so when we think about that autonomous nervous system, it's going to be kind of broken up into two major divisions. We have the sympathetic division and the parasympathetic division. The sympathetic division is that's going to be that fight or flight type of response, and the parasympathetic division is going to be responsible for. Let me write that up. And the parasympathetic division is going to be that one is going to be kind of rest and digest. Okay. And you kind of think about this, right? Think about you as an individual and think about how it pertains to you. So let's say a bee. A bee is coming up, big old, gigantic bee flies towards your head. What are you going to do in response to that?
Well, I leave it alone. Some people just, my son would try to smack it out of his face.
As a center, what would you do?
Oof. So it could go away.
You try to smack it away?
No, I'll probably stay still and try to make the bee like fly away by itself, so it
Okay.
not flower.
Okay. Okay, but when you're just chilling and you just had a really great meal, you just watched your favorite show and you're like, it's after a long day of work. So what are you doing in that case? How are you responding to that?
Well, if I see it coming, I think I probably like try to dodge it. But if it came out of nowhere, then yes, definitely I would probably try to like smack it off.
Yeah. Okay. Now, when you do this, or when you have an event that kind of works you up or gets you really scared, and you're like, oh, have you ever had an event like that just kind of worked you up and get you really scared?
Yes.
Yes.
Okay. So what happens in terms of, did you feel hungry? Like during that time period?
No.
No, you didn't feel hungry. Did you even like, maybe if you were in the middle of getting food, or you wanted to get food, or you were just ready to eat and this happened right before that, could you eat right afterwards? Did you eat immediately afterwards, or did you have to settle down before you could grab that meal?
Settle down.
You have to settle down, right? Okay, so in the case of the sympathetic fight or flight response, so in the sympathetic division, when we kind of get that system or that division worked up, what happens is it starts to change other things. The things that they've noticed is that we start breathing, heavier and stuff like that. So those cardio muscles and stuff like that, they really start to work, right? They start moving because we're ready to fight or flight. So either we're gonna have to run really fast, or we're gonna have to stand here and fight whatever it is that we're gonna have to fight, right? So during that time period, we also start sweating. You get all sweaty and everything like that, and just, And so in that case, we're starting to, our muscles are going to be contracting, our body's going to start overheating and stuff like that. Hey, we need to be ready to sit there and fight. So where our digestive system is going to shut down. So we're not going to be able to absorb those nutrients. Even though we're breathing heavy and things like that, all of that is going to be sent towards our skeletal muscles to sit there and prepare for that fighting, rather than in our digestive system, it's not going to be used for that process. So we're going to be using some of that glycogen that we have, and then we're going to be using that oxygen to help sit there and bring that bonus of the glycogen down. Okay. Are you with me so far?
Yes.
Yeah.
See where I'm going with this? Okay. So if we go ahead and so these are basically the connections of the sympathetic divisions of the autonomous nervous system. So it kind of gives you an idea where everything is located and what it's sending signals to. So if we take a look, we have neurons that are going to be on the lateral horn of the spinal cord. They're going to project to the chain ganglia on the other side of the vertebrae column, or they're going to be projected to the correlated ganglia on the anterior vertebrae column in the abdominal cavity, and then and again, you have a much better picture in your book, so you'll kind of get an idea of the projections and the projected components. And then if you take a look at the axons from the ganglionic nerves, so then they're going to be projected to their target effectors. So, and that's going to be the organs that we're going to be looking at. Okay. Come on. Okay, so this kind of gives you an idea of how the connections are formed, right? And the signaling. So basically, we have so the axons from the central nervous system, the central sympathetic neuron in the spinal cord is going to be projected to the peripheral in different ways. So one of the ways is that you're going to have either the fiber can be projected out from the ganglia at the same level, and the synapse on the ganglionic neuron. And so that's going to be kind of shown here. So if you kind of hard to see, but basically you have your synapses, you have your projection here, and then you have your synapses occurring at this point right here. Okay. And then if we take a closer look, so here's the spinal cord. So here we have it extending out here. It goes to the spinal nerve, and then from the spinal nerve, it actually snaps at this point right here. Okay, So, and then from there, it goes to the targeted sector. Okay, and so this guy is considered going to be your right ramus communicon. And then in B, here's B, you're going to have basically a branch can basically project from a more superior or inferior ganglion in a chain. So in this case, you're going to have your central neuron synapses within a more superior. Okay, so again, it's really hard to see. It's a little clear, not too much clear. You have your neuron basically branching and it's going along here. And so you're going to have your synapses here, along here, along here, along here. And then these guys are going to be hitting those effectors, okay, from here on. So you're going to have your synapses along this part. Okay, so, and here's a little bit better picture of it. So you have, again, the axon extending from that nerve, the spinal cord to the spinal nerve. And then it continues that extension and it goes up to your ganglion, where you actually will have a synapses occurring there. Okay, and then that's going to go to our effector. Down here, the third one. So basically, you're going to have again, it's going to be the branch is going to be projecting from the white ramus communicon. So in this case, you're going to have these guys here, and then here you're actually going to get like a branching structure. So it's going to be protected by a chain, but a little chain of little synapses. Okay. So instead, it's gonna be projected through one of the sympathetic nerves, and then that's going to be nerves to a collated ganglia or the adrenal medulla. Okay, so in this case, here's a better picture, but they're not showing you the adrenal medulla. It's not being shown in this picture, but the other one is. So basically, we have the little nerve. It goes here to the sympathetic neck nerve, and then this is where you're going to have that expansion or your response. And so, and that's going to be in this case the prevertebral ganglia. Okay, and let's see what else. So in the case of the parasympathetic divisions of the autonomous nervous system. So the neurons from the brains, and so we're looking up here. Or from the lateral horn of the sacral spinal cord is going to be projected to the terminal ganglia near or within various organs of the body. Okay, so the distance that these guys are going to be projecting, there's going to be a shorter distance to the different organs, the target organs. So if you kind of take a look at it, so all of those guys in blue, right? So we have the cranial nerve X, and that basically is going to be connected to those inner organs. So that's going to be like your heart, your larynx, your stomach, all the way down to like your intestines. The cranial, I guess that's nerve 10. The cranial nerve 9. So that's going to be to your mucous membrane and your parotid gland. And so like your cranial nerve 3 is going to be attached to your eye. And so the distance that these guys are traveling are going to be smaller, so or shorter distances than you had in the case of the sympathetic nervous system. Come on now. Okay, and I'm actually going to stop here, and then we'll pick this back up on Monday. And so you guys have kind of already seen this anyway, but we're going to talk a little bit more about those neurotransmitters and some of the different types and some of the effects that we have under these circumstances. But like I said, we'll be picking that up on Monday, and we'll finish up chapter 15 then, 15 and 16. So if I remember correctly, correct me if I'm wrong, so that means that we have 3 weeks. We'll have 4 weeks left after this week. Is that correct?
Yes.
Yes.
Okay. So 4 weeks left. And during the last week, I'll give you guys the whole week, but the last week is going to be covering a couple of chapters, and we're going to kind of zoom through that last week too. And your final is all going to be multiple guess, no short answer. So to kind of sum it up, your final is going to be focused primarily on that last bit that we covered. And you technically have one more exam to take after you guys finish this exam. So in 2 weeks, you're going to have another exam, and then you're going to have your final. The final is going to cover the last. It's going to be, you're really going to be made up of 3 chapters. Not 3 chapters, 4 chapters in theory. So next week, we're going to be going over chapters 14, 15, 16, and 17. It's going to be very condensed. So I'm going to try to give you the major components. And you're going to have one more lab to do with the fish. I was going to have you guys do 2 labs with the fish, but I'm going to break it down to 1. We're going to try to get it all done in one setting, because we want to do the stimulus of the alcohol response, stimulus of the eyes, and then also the response of caffeine. See how all of these different components together affect the fish's response. And so we're going to be looking at the nervous system in that regard, and we're also going to be looking at the skeletal muscle systems. And then, of course, the respiratory system, because we're going to use breathing as well. So I've kind of had to think it through to try to get it all concise, because we're going to basically get our fish a little cross-faded. You know, it's going to be a little drunk, and it's going to be hyped up on caffeine.
Hopefully it makes it. It sounds like too much for it.
Yeah. Okay, so yours is 3 grams. Okay, so good. So I figure so far of all the fish that I've seen, Alejandro's fish is the biggest and probably weighs the most. And so we'll kind of use his weight as our upper range. So yeah, he'll make it. That's why I'm trying to figure out how much should I give it to get the effect that we want to see without, you know, killing it, you know.
Yeah.
So, but if you think about it, caffeine and alcohol are kind of active to each other, right? One is a stimulus and one is a depressant, right?
Mm-hmm.
So, so like for instance, have you guys had, what is it called? We call them bombs. So we call them like
a Jaeger bomb.
A Jaeger bomb, yeah. So like a Jaeger bomb. So it's Jaeger and Red Bull.
That's too harsh.
Right? So you're in this sense, you're getting it hype up, and then you're also putting it down. So, but we want to try to see the effects of each one individually. So we need enough time to give, okay, so first we're going to give them this, then we're going to give them this, and then we're going to give them this, right? And so I need to try to figure out what is the best way of doing this without having it all done at once, you know. And then in addition to that, you're going to play with you guys have the glow stick still?
Yes.
Okay. And then you're going to have the glow stick, and then you're going to have the response to that. So the presence of the alcohol, in the presence of the caffeine, you know, in the absence of the alcohol, in the absence of the caffeine. So you're going to look at it that way and see how he responds to that light stimuli. It would be kind of nice if we had 2 fish, right? Because then you can do that. But it would be kind of good to do it with 1. I'll figure it out. But we have 2 experiments to do each week. So maybe I'll break it up. I'll have you guys do on Monday, we'll do like the alcohol. And then on Tuesday, we'll do the caffeine. And then we'll follow up with caffeine and alcohol at the end of Tuesday.
Okay.
Yep. And did I give you guys a packet of glow sticks, or did I give you 1 glow stick?
I believe it was just 2 glow sticks.
I gave you 2 glow sticks.
Yeah.
Okay, so then that's good. Okay. Okay, so I will see you guys next week. You guys should have a lab that's already online, the Physio Exo Lab.
Mm-hmm.
And so you guys should be good to go there.
Okay.
Okay.
Sounds good then.
Okay. I will see you guys. That is a short time.
Have a good rest of your week, Dr. Henry.
You too.
Day and week.
I guess.
Bye.